Strep pneumoniae can be broken down into strepto, which means chain, coccus, which means round shape, and pneumoniae, which refers to the fact that it causes pneumonia. No surprises there. So strep pneumoniae are round bacteria that tend to grow in chains, usually in lancet-looking pairs called diplococci. They're the most common cause of community-acquired pneumonia, meaning pneumonia acquired somewhere other than the hospital. Okay, now, a little bit of microbe anatomy and physiology. Strep pneumoniae has a thick peptidoglycan cell wall, which takes in purple dye when gram-stained. So this is a gram-positive bacteria. They're non-motile and don't form spores, and also they're facultative anaerobes, meaning they can survive in both aerobic and anaerobic environments. Finally, they're catalase negative, which means they don't produce an enzyme called catalase. Okay, now, when cultivated on a medium called blood agar, strep pneumoniae colonies cause alpha-hemolysis, also called green hemolysis, because they produce hydrogen peroxide, which partially oxidizes the initially red hemoglobin in the blood agar to create green methemoglobin. Other Streptococcus species, like strep viridans, are also alpha-hemolytic. So an opticon test is done to distinguish strep pneumoniae. That's where an opticon-containing paper disc is placed on a plate of blood agar that has been inoculated with the bacteria. With strep pneumoniae, the growth of the bacteria is inhibited in the zone that surrounds the opticon disc, meaning that strep pneumoniae is opticon sensitive. On the flip side, strep viridans are opticon resistant, meaning that there's no inhibition of growth around the opticon disc. Another test that can be used to distinguish strep pneumoniae from other alpha-hemolytic streptococci is the biosolubility test. The biosolubility test can be done in a test tube, or more commonly, by dripping drops of bile on bacterial colonies. Now, strep pneumoniae is biosoluble, meaning that the bile salts will promote the lysis of the bacteria, whereas all other alpha-hemolytic streptococci are bile insoluble, or bile resistant. Now, strep pneumoniae has a number of virulence factors that are like assault weaponry that help it attack and destroy the host cells and evade the immune system. So first, strep pneumoniae is encapsulated, meaning it's covered by a polysaccharide layer called a capsule. The capsule has pili and fimbriae, which are hair-like extensions that help it attach to a host cell. Once attached to a mucosal surface, like in the nasopharynx or the middle ear, strep pneumoniae can multiply and produce biofilms. A biofilm is basically a layer of goop-like material made of exopolysaccharides or EPS, within which strep pneumoniae bacteria live and reproduce. Comparing a biofilm to strawberry jam, the seeds would be the bacteria and the rest of the jam would be the EPS. The bacteria in the biofilm hide from the host's immune system and antibiotics, and even exchange resistance genes. Sneaky. Another virulence factor is pneumococcal surface protein A, or for short PSPA, which inhibits complement activation. This way, strep pneumoniae avoids the opsonization and eventual destruction of the bacteria. As if that wasn't enough, strep pneumoniae also produces toxins, and the most important one is IgA protease. IgA protease destroys immunoglobulin A, or IgA, which normally binds invading bacteria so neutrophils can destroy them. That's like a computer virus taking over the antivirus software. The next virulence factor is autolysin. Activation of this enzyme causes the breakdown of the bacteria and the release of its internal components. Released internal components further activate the host's immune system, thereby enhancing the local inflammation and destruction of the host tissues, like alveolar capillaries and pneumocytes. Now, one of the components released from the bacteria is pneumolysin, which is another virulence factor. Pneumolysin is a toxin that binds to cholesterol in cell membranes and forms pores, which eventually lead to cell lysis. And here's the most interesting part. Strep pneumoniae can actually peacefully colonize the nasal cavities and sinuses, where it doesn't do any harm so long as the immune system keeps them in check, restricting their growth and preventing them from spreading somewhere else in the body. Problems arise in individuals with weaker immune systems, like infants and the elderly. Other immune-weakening conditions include an HIV infection, diabetes, malignancy, or alcohol abuse. Additionally, some toxic compounds in cigarette smoke can weaken the local respiratory defense mechanisms, making individuals more susceptible to strep pneumoniae infections. Strep pneumoniae can cause a number of infections. One example is rhinosinusitis, which is when the mucous membrane lining of the nose and paranasal sinuses gets inflamed, resulting in fever, facial pain, and headaches. Strep pneumoniae can also invade the eustachian tube and cause an infection of the middle ear, or otitis media, which causes pain and an earache. Chronic otitis media can spread to the mastoid antrum behind the ear, where it causes mastoiditis. Finally, from the mastoid antrum, the infection can spread to the cranial cavity, which is one way strep pneumoniae causes meningitis. However, the most common disease caused by strep pneumoniae is pneumonia. Strep pneumoniae sticks to the alveolar cells, the pneumocytes, and releases pneumolysin, which leads to the destruction of the pneumocytes in alveolar capillaries. Pneumonia causes fevers and chills, chest pain, and a cough. In severe cases, strep pneumoniae can cross the alveoli capillary wall and enter the blood, resulting in bacteremia. Eventually, bacteremia can cause serious problems, like sepsis, which can lead to hypoperfusion of vital organs. Sepsis caused by strep pneumoniae is particularly common in people with spleen problems, because the spleen plays an important role in immunity, especially against encapsulated bacteria. So strep pneumoniae infections are typically worse in individuals who have undergone a splenectomy, which is when the spleen is removed, or in those with sickle cell disease who have functional asplenia. Once in the bloodstream, strep pneumoniae can cross the blood-brain barrier, invade the central nervous system, and cause meningitis. With meningitis, people develop a fever, neck stiffness, and a headache. On rare occasions, when the walls of the paranasal sinuses are very thin, strep pneumoniae can get into the cranial cavity and cause meningitis. Strep pneumoniae can also cause pneumococcal endocarditis, forming clumps called vegetations that damage the valves. It can also infect the pericardium, resulting in purulent pericarditis, which is where pus collects in the pericardial sac. Strep pneumoniae can also spread to a joint, resulting in septic arthritis. Or it can cross the blood-brain barrier and cause meningitis as a complication of bacteremia, rather than invading directly as part of a primary disease. Once it has crossed the blood-brain barrier, it can also cause an intracranial or spinal epidural abscess. Finally, strep pneumoniae can spread from the bloodstream to the peritoneum, causing spontaneous bacterial peritonitis. This is more common in individuals with cirrhosis who develop ascites or too much fluid in the peritoneal cavity. Diagnosing a strep pneumoniae infection relies on cultures from biological samples like pus, sputum, cerebrospinal fluid, or peritoneal fluid. Alternatively, PCR can be done to detect strep pneumoniae DNA. Individuals at risk of strep pneumoniae infection should get vaccinated. This includes young children below 2 years, as well as people over 65 years old. Other risk groups include those with chronic diseases, such as diabetes, HIV, cancer, and most importantly, those with sickle cell disease and those with functional asplenia. Currently, vaccines against strep pneumoniae are divided into two main groups based on their mechanism of action. The first one is the pneumococcal polysaccharide vaccine, also known as PPV-23, because it contains 23 purified capsular antigens, and this one is mostly recommended in adults. Another category is the pneumococcal conjugate vaccine, or PCV-13, which contains the capsular polysaccharides specific to 13 strep pneumoniae serotypes, bound to a non-toxic recombinant variant of the diphtheria toxin, called CRM197, which serves as a carrier protein. PCV-13 is recommended in young children. Finally, treatment for infections with strep pneumoniae requires antibiotics. In the classic first line of treatment are beta-lactam antibiotics, like penicillins G and V, or cephalosporins. However, strep pneumoniae has been progressively developing resistance against penicillins and its derivatives such as amoxicillin and ampicillin. So alternatives include fluoroquinolones, the combination of amoxicillin and clavulanic acid, as well as third-generation cephalosporins like ceftriaxone and cefotaxime. Ultimately, vancomycin and linezolid can be used. Alright, as a quick recap. Strep pneumoniae is a gram-positive round bacterium that lives in chains. They are non-motile, non-spore-forming, alpha-hemolytic, optic-insensitive, and bile-soluble. These bacteria are found asymptomatically colonizing the nasal cavities and sinuses, but they can take advantage of a weakened immune system, especially in very young or very old individuals, or those with diabetes, HIV infection, cancer, splenectomy, or those with sickle cell disease. They cause diseases such as rhinosinusitis, otitis media, pneumonia, and meningitis. The treatment of infection with strep pneumoniae involves the use of penicillins, but in the case of resistant strains, fluoroquinolones, third-generation cephalosporins, and vancomycin can be used.